Namaste. I am Save the Frogs founder, Dr. Kriger. This is the South Asia Meetup. South Asia Meetups take place last Saturday of every month at 7 p.m. India Standard Time. We've got Save the Frogs India founder Sarbani Nag here, and we've got Dr. Karthik Vasudevan planning to give a presentation, though, with current internet issues. So we're just going to do this. Everybody out there, if you would like to turn on your video, you're welcome to turn on your video. If you'd like to turn on your audio and introduce yourself quickly, concisely, like where are you, what's your background with amphibians, if any, and tell us what's going on. I'm currently in Tolima, Colombia, and I have been saving the frogs since 2003 when I started my PhD research in Southeast Queensland, Australia, on the ecology of chytridiomycosis in Eastern Australia, amphibian diseases. I founded Save the Frogs in 2008 as the world's first and only public charity dedicated exclusively to protecting amphibians. And I visited India and Nepal in 1997 and returned to India in 2017, 18, and 19, Bangladesh in 2018, and gave various presentations on amphibian conservation throughout India and Bangladesh. Save the Frogs has been supporting amphibian conservation throughout South Asia for a very long time. I know that India has been involved with Save the Frogs Day since the very first Save the Frogs Day in 2009. Everyone out there, please mark Save the Frogs Day, the 17th annual Save the Frogs Day, April 28th, 2025. Mark it on your calendar and plan to take part. You can organize an event in your community to educate people about amphibians and inspire them to protect them and educate them about amphibians. Save the Frogs has given out a large number of grants to amphibian conservationists in India, Pakistan, Bangladesh, and Nepal. And we hope to continue doing that. I know that we have some grant winners here on the call right now. So perhaps one of you grant winners out there could introduce yourself. And if you won a grant this year and held a Save the Frogs Day event, maybe you can briefly tell us what you did, what you learned, provide any advice for anyone out there. Meanwhile, everyone out there, I encourage you to apply for a Save the Frogs scholarship. You can go to savethefrogs.com/scholarships. If you are dedicated to amphibians, that will get you a Save the Frogs membership and full access to Save the Frogs Academy, where we have many courses, online courses on amphibian conservation, including, as you likely saw when you registered, we have a course for India, Nepal, Bangladesh, and Pakistan, and many other courses. So go check that out, savethefrogs.com/scholarships. If you're interested in amphibian conservation in other parts of the world, perhaps you could apply that knowledge to your part of the world. In exactly 3 hours, we have a Save the Frogs Africa meetup focused on African amphibian conservation. And you can go to savethefrogs.com/Africa to register for that if you would like to attend. Those also happen the last Saturday of the month. If you have questions,
yeah. Should I continue with the introduction?
If you guys are ready to begin, then yes.
Okay, so I met Dr. Kartik Kriger in Kolkata. That was back in 2019, and we did a seminar here And after that, I applied for the Save the Frogs Day grant, and I won it. And since then, the journey is continuing. And later, I registered Save the Frogs India as a trust so that I can do all these things. Conservation of frogs across India, and Karthik has shared the PPT with me, so I'm trying to sharing it with me too. So
Are you able to see the
yes,
presentation?
yes. Your screen is visible, sir.
Okay, great. So I'm really, really sorry for this glitch. I've never, this has not happened in the age where we are doing so many online presentations. This is the first time I'm facing this. Anyway, so thanks, Sarbani. So I'll just start with the presentation and yeah. Is it on a presentation mode now? Can you see
I
the...
see, everything looks good.
Okay, so fine. So I was telling you that earlier this year we had this publication come out on global amphibian assessments that highlighted that amphibians were declining, continue to decline, and the drivers have probably changed a bit, but the trends in their population continues to be on the decline, which is of concern, and we need to reposition ourselves, reorient, and also investigate in ways that we can understand these animals and respond to the situations they might get themselves into because of the changes that we are causing to the environment. And India is no exception to it. And among the areas where many threatened species occur, India comes out very prominently. And India and its neighboring countries share many of the concerns that that are common, and the land use and the aspirations of the people are similar, and the changes that they cause to the land and the freshwater resources are pretty much similar. And you have the same region experiencing vast changes due to climate change presently, which is also one of the important causes for these declines. But we really need to spend a lot of effort to understand what is the status of these amphibians in these tropical areas, and how do we even know if they're doing well or if they're declining or they're into some problem. So the situation that we have today is that among the top 10 threatened genera we have in the Indo-Malaya region, we have three genera which are found in India, and two of them are anurans or frogs, and one is a salamander. In India, you have salamanders, caecilians, and frogs. And the characteristic feature is that all of them show high levels of endemism, which means um, these animals remain in these areas and have narrow distribution ranges. And this prompts us to investigate how do they survive with such narrow distribution. And this is the biogeographic classification of India. And you can see that uh, India might look more homogeneous in terms of the land features, but characteristically there is a mountain range running along the south from north to south. This is a mountain range called the Western Ghats, and there is a mountain range running north on the Himalayas, starting from almost the western end of India, and goes down till the eastern end of the country. This chain of mountains obviously passes through Nepal, Bhutan, and then ends up meeting another hill range, which continues south, going into Southeast Asia. And there is an abrupt break, and there are submerged mountains in the ocean in the Bay of Bengal called the Andaman and Nicobar Islands. The Andaman Islands is a continuation of the same mountain range that runs in the southeast, but they're sunk under the sea. So you have these islands which are which are having a land bridge to the mainland. And further south, you have the Nicobar. They are truly oceanic islands. They are volcanic in origin, very different geologically in terms of the time when they have emerged. They are very similar to the islands of the Indonesia, and there is a large gap between these chain of islands. And the mountain range in the mainland of India, which is the Western Ghats, also is fairly contiguous. And this prompted us to investigate what's happening in the Western Ghats. This is how I started my work on frogs way back in 1990s. And we focused on two sites because forest fragmentation was a major issue, and it continues to be so. Prime evergreen forests are being converted into plantations of tea, coffee, cardamom, and other cash crops. And these were historically fragmented, but even now many of the forests are being converted into cash crops, different cash crops. So when you get into these forests, it's a prime evergreen forests have lots of leaf litter, and you have to search for these frogs by sifting the leaf litter. And we placed quadrats or demarcated areas, and then sampled within them by turning the litter and seeing the animal move, and then capturing them and measuring them, or photographing them. And this was prior to the the taxonomic boom that happened in 2004-5 and continued on till 2015. So we knew that many of these species were undescribed even then, but it would require a lot more work to describe all of them. But our effort was to understand how many of these identifiable frogs are there. And another approach that we used was to walk through the streams in the night and look for frogs. 100-meter stretch of the stream was walked, and then we would count the frogs and identify them without capturing them. So in the night, when you flash the light and walk through the streams, amphibians seem relatively undisturbed, and you can spot them easily. So when we did that for the first time, we got fairly good estimates of the numbers of these frogs. Some of them are, some of these numbers typically show that we can count them, we can assess their populations, we can look at their trends. Though the names of these frogs are restricted to the genus because of the taxonomic uncertainty that was associated with it. But roughly 1 hectare of the forest of the riparian zone in the Western Ghats had about between 250 to 400 frogs. This is a very important number. I'll come back to it when I talk to you about other places. And this tells us that this range also tells us that the forest floor is quite heterogeneous, or the habitats for the frogs are pretty heterogeneous. And it's not that every place will harbor as many frogs as some other better site would. So this is very important. Another important finding that we got was that there is a lot of turnover of species from one area to another, and this is very distinctive when you move from one drainage to another. So there is almost 80% change or 90% change. I would say if you do a very thorough taxonomy, it might be even more than 90%. That different hill ranges have totally different assemblages of frogs. So this means that protected areas need to be distributed rather than single large protected areas. So if you need to conserve amphibians, you cannot focus on one small piece of land. You'll have to spread your effort across the Western Ghats. So another similar effort, I don't think these images would move, but I will just explain. They are just nice pictures of those habitats, but the animation is probably not working in this slide. So basically along the Western Ghats, you have high elevation areas restricted to the south, but you do have some peaks going up north. And this means that there are two bounds or extreme geographic limits of the frogs distribution in the Western Ghats. One is the Arabian Sea here, and the other is the peak. So all the species have to distribute themselves along the slopes, and that's the space they have. And typically, frogs are found everywhere along this elevation range. And we try to look at what is the pattern of species richness as we go up. So these are two areas. This is at 8-degree latitude, which is in the southernmost tip. And this Kudremukh is in the northern, in the central Western Ghats, but at least separated by 8 latitudes from the southernmost part. So you can see the pattern in species richness varies significantly. And these red lines that I have drawn, something like a curve, primarily tells you that that is what you expect if you were to assign species ranges randomly between the two bounds, that is the sea level and the peaks. So the pattern is slightly different, but what is the take home here is that two areas, one is in the lower reaches and the other is in the higher reaches or the peaks of the mountains, typically have more species than you expect. And they are the most impacted also. Almost all the low elevation forest habitats are gone. They have been removed, they have been changed for cultivation or human habitation. And the mountains are now changing so rapidly with climate change that the precipitation regimes are shifting, and that is going to cause major problems for amphibians, both on the top as well as on the bottom. So if I just show you the parallel between the Western Ghats and the Andamans, they are very similar in terms of the range of latitude that they cover. And the two sites here, which is in the Andaman and Nicobar chain, the Andaman group of islands and the Nicobar group of islands, were important to investigate for the same reason that I told you, that the turnover of species is very high in the mountains. And I also told you that the Andaman chain of mountains are actually subducted mountains. That means the mountains have sunk under the sea. They are linked to the mainland chain of mountains, but they have sunk under the sea. So, which means you would expect the same pattern that you see in the Western Ghats here as well. But we were quite surprised by what we saw when we went and looked at these islands. So these islands are typically very, very beautiful islands. They have very large evergreen trees. And a student who worked with me, S. Hari Krishnan, He is now championing an amphibian recovery project in the Western Ghats with the Wildlife Trust of India. And he did an amazing job of documenting the diversity in these islands. So this is how the islands seem as you approach them. You can see that these trees have all died out. This was a photo taken much after the tsunami in 2004. So there's a lot of dieback of trees because of saltwater incursions into the island. So we did some surveys post tsunami as well. We had some surveys done before tsunami. So we could compare and say that, I mean, we had some, it was very difficult to reach the islands post tsunami, but we had some evidence that amphibians had gone from many of the habitats that were originally occupied, but interestingly, they could colonize, they could come back very quickly. And that was very reassuring. And this is as you enter the island, the forest is called the littoral forest. That is that is like the testing ground for any species which enters an island. And if they survive the littoral forest, they will go inside and occupy the montane or the evergreen forest inside. So the inside of the forest is fairly protected by this littoral forest, which takes them much of the beating by winds and storms and everything. And the inside of the forest is pretty much intact. So when we did this work, we also investigated the mountainous habitat for the reason that they are so similar to the mountains that you find in the Western Ghats. But these mountains are very short. They are just 500 to 800 meters tall and very small mountains. But typically, they look like mountains which are over 1,800 meters in the Western Ghats. The trees become dwarf. There is always some mist at 500 meters. It's much cooler there than 500 meters below, which is at the sea level. So there is a stark climatic change when you come up even 500 meters in these mountains in the Andamans. So we looked at body size and evidences that there is some structuring in the community of frogs, whether there is some competition or, what is the assemblage of frogs there, and what is their population, and how many islands they occupy. Usually you expect that species that have more numbers tend to occupy many islands, which was not really the case. We didn't find any such pattern. And also we didn't find any pattern to suggest that there is a modal body size where there might be some competitive interactions and speciation among the frogs. That is also corroborated by the fact that there are fewer endemic species in these islands. Some of them, for a single island, there are very few endemics, but together in the Andaman archipelago, there are many endemic species. So we could describe one new species of toad, which is a bush toad, an arboreal toad found in the montane forest, and now also found in 5 or 6 different islands. And something as common as this, or found in the largest island and other islands associated with it, having to find them in 2015 is quite a significant thing, because it just shows that we haven't looked at these areas carefully. And the important thing to note here is this: the species epithet, Beryet, Beryet, is a derived name from. Actually, it is the name from the Great Andamanese lexicon, or the language. The Great Andamanese are a group of people who lived in the Andamans, the native people of the Andaman Islands, and their language is now almost extinct. But a linguist from University of Delhi carefully documented all the words, and they had a word for a bush frog, and there's no other bush frog in the Andamans. The Andamanese knew these frogs much before us. It's just that they had a different name for it, and we were formally able to describe it for the international community with a binomial. So the threats for these frogs now, through our work on chytridiomycosis in this part of the world, we know that it is prevalent in these frogs. We don't know what happens when they get infected by this disease, which is a global pandemic. Forest cover loss, littoral forests and mountain forests are getting more and more disturbed and being converted into other land uses with large interests in promoting tourism in these areas. Then you have a constant threat of
सही चलना पे
is there a question from someone?
No, I guess you should continue, and other participants
Continue.
should mute themselves. Yeah,
Okay.
please continue.
So there is a constant impact of stochastic events like tsunami. It happened in 2004, and this can happen anytime in future. And the good thing is that we know that frogs are more resilient to these stochastic events, and they seem to recover from it, which is a positive.
Can you explain a stochastic event for the people who may not know?
Sorry. So the stochastic event is one which you don't anticipate or something which you can predict. It's a event that happens by chance, and it can be a event that completely removes a species or its population from an area. It could be landslides, floods, tsunamis, earthquakes. All these are stochastic events. Though we wish we could predict them, they don't happen with much warning. So they just wipe out a species or a population, and they're natural events. They're not man-made. They're natural events. So clearly, these frogs are adapted to these natural events, but on the contrary, they are not adapted to invasive species, which are introduced by humans. And there are many invasive species in these islands, from herbivores to carnivores, and they continue to impact populations of frogs, some of which we could document, some of them we don't know what are the impacts. So I'll give you the case, though I don't have a presentation to show. One of my students went on to do his PhD on the Indian bullfrog, which was introduced into these islands, and it is a carnivorous bullfrog. It feeds even on frogs, snakes, lizards, everything that it finds on its way, and it has caused a huge impact in these islands. The impact on frogs particularly is not well evaluated, but his study revealed that just a single invasive frog, can be a big impact in the freshwater ecosystem. So you have the same situation panning out for frogs with respect to the chytridiomycosis. Globally, this pandemic has devastated several continental regions, the Australia, North America, South America, Europe, and other species of the fungi also impacts salamanders. It's called chytridiomycosis, Bacteriochytrium salamandri vorans, and Batrachochytrium dendrobatidis, which is abbreviated from now on as BD, is what I'm going to talk about. It's basically a fungi, which is very distinct group, which has motile zoospores. Zoospores are basically seeds of this fungi, and they disperse after setting their spore onto the skin and then dividing into many zoospores, and then the zoosporangia develops a tube, and then they release the zoospores into the environment, and they are obligate pathogens of frogs. Obligate means they don't infect other species, but we know that they do occur on other organisms like freshwater crustaceans and the skin of wetland birds. So they can disperse through the environment and then occupy, infect another frog, and then spread the disease. And when they are infecting the frog, they cause a lot of damage to the skin. The skin in the frog is a respiratory organ, and also an important organ that regulates the water retaining capacity or the salt retaining capacity of the body. So they function like the kidneys of our kidneys. So the skin absorbs water from the environment and is able to keep the body in a condition which can allow them to perform all their activities. But when the infection happens, the frog generally becomes very lethargic, and it dies very quickly after the salt composition in its blood changes due to the lack of osmoregulation through the skin. So this fungi was recorded from almost all parts of the world, and we didn't know much about what's happening in India. So we started the work in 2014, and we have published a few work through our continuous monitoring of this disease in different parts of the country. And I'll show you some slides on this. And the situation that was there, this is prior to 2009, I think, the slide on the picture on the left. And then you have the situation on the right, where we did this survey throughout India, and we found BD occurring almost everywhere in the country. And there were roughly 40 different haplotypes of the fungi. Haplotype is basically a variant of the fungi. All of you now must be familiar with variants because of the COVID infection, and we know that there were so many variants coming from time to time with different levels of virulence. So this is basically what is happening to the frog populations here. It's a blender of different pathogenic variants of BD that occur here. And you can see that this really needs a very serious investigation. Although we don't report any morbidities or deaths of frogs, but it's called as a cold spot now of infection, where the pathogen is considered to be enzootic. Enzootic means that the pathogen is stopped becoming virulent and not causing mortality, but certainly is present in the population. So the take-home message from our initial investigations was that there is a large number of frogs which are infected. We need to have better ways to detect them, because the conventional methods to detect them were not working for the strains that were involved in infection in India. So we had to develop some better assays, better detection assays, and there is some host mechanism that is playing a role in reducing the infection loads, which I will talk to about in the next few slides. And basically the situation in India presents a new window of opportunity to explore what happens after the pathogen sort of reconciles to survive in the environment by modifying the host responses and continue to infect the host repeatedly. So after we developed a new assay to detect BD, basically the assays that were available were showing a lot of false negatives. That means we did not detect chytrid while there was chytrid using the old assays. So we had to develop a new assay, and that detected chytrid. And for the same samples that we used the old assay, we had 2 positive of the 139. And when we did in 2018 with the improved assay, we got 82 positive. So it just completely shifted the knowledge about what's happening in these areas. We always thought that cold spots had low levels of prevalence, and thereby the frogs were not getting affected. But now we know that most of the frogs have chytrid, but they are just somehow keeping it at bay, not to allow them to completely run over. So, and we also for the first time saw that many caecilians were affected, and even salamanders had BD. So BD was in the environment, and it was also shifting hosts. Though it has been recorded in salamanders, caecilians, there is only one record so far. So we started to focus on investigating the cold spot more seriously, and work in a small area, well defined. We selected 5 different streams, segments, and we started to investigate what was happening there by performing a capture mark recapture study there. So this area, typically the Western Ghats receives the southwest monsoon. It arrives in the southern tip at the month in the roughly around the 1st of June every year, and then moves northwards. So by May, the hydro period starts. The blue bar that you see on the months here indicates the period where there is heavy rain and continuous rain. And the black graph that you see here is the amount of rainfall. And we indicate this period as the pulse period, or the entire drainage is flushed out with water. And because of the heavy precipitation, over 2,000 millimeters of rainfall on a few days of the year can cause a complete swelling of these rivers, and nothing can, even we cannot ford over these rivers, we cannot cross them. So the frogs are all hiding during this time, and they are not in the streams. So once the rainfall subsides, you get an opportunity to get into the stream. The frogs start to become active, and they have a very short period after that, roughly 2.5 months, where they have to do everything that they can in terms of breeding and moving from one place to another, finding better spots to lay their eggs, the tadpoles to develop and metamorphose and come out, because come end of December, you don't have water in the streams. Everything dries out. So this is a very stark habitat where, though it receives over the 4,000 millimeters of rainfall in a year, it becomes dry for at least another 7 months, 8 months of the year. So this is, if you see the dry period here, hot dry period, February to June. This is how the same stream looks like. And you have in the monsoon, almost the entire stream is in spate. And then slowly the water spread reduces from the wet period to the dry period. And then finally it dries out completely. So we recorded in all 27 species in this area, and we could follow a few more abundant frogs or stream dwelling frogs in this study. So essentially, it's the same method that I mentioned, like a visual encounter survey, but here we capture the frogs and mark them. And marking involves inserting a small tag under the cutaneous layer of the skin, and this tag remains under the skin of the frog. And the frogs are caught in these individual Ziploc covers, and then they are read using a reader, and it gives a unique number that identifies the frog. So we used a very low payload of the tag, which doesn't cause much damage or inconvenience to the frog. So this is the data that we have over the last 5 years. We have the 6th year data as well. So we were able to find that the population is relatively stable. The line that connects the dots is the population graph. And you can see that the population tends to be stable. There is a wet period in 2021, which we did not go and survey. We went and surveyed only the dry period. So there is a break in the graph, but essentially it comes back to the same numbers. And the bars are basically indicating the number of days there were rains in the study area, and the colors indicate the amount of rainfall during that period in this color code, which is presented on the right side. So basically the rainfall pattern changes every year. It's not the same. The wet and dry periods have different number of rainy days between the years, different amount of rainfall between the years. But what is consistent is that the number of there is some rain during the dry period, but there is also a substantially large number of wet days during the wet period. And there is very high rainfall on some of the years, not much on some of the years. So this is a pattern that the frogs don't anticipate, but they seem to somehow adjust. This is just one species, which is the skittering frog, and they seem to find ways to come to the streams in the same numbers to these and occupy them during the period that there is water in the streams. So with respect to chytrid, we found close to 75% of the frogs had infection, and many of the endemics like the Amboli leaping frog, the Jog night frog, and the Maharashtra golden back frog, all of them had high levels of BD in their population. So this is just prevalence. This doesn't mean that they have high loads of the pathogen. Usually the loads were pretty low, and I just present the results here of the loads on the right-hand side, where you can see that almost always the loads were a little higher during the wet period, and it just dropped a little bit during the dry period. So this pattern was very consistent, and we are continuing to monitor the site. So essentially what we found was that frogs that come on the first year don't reappear on the second year. Very few of them reappear in the same site, and there is a total turnover of population every year. So there are no symptoms. There is high number of individuals which show infection in a population. There are no deaths. So this is the situation that exists in a cold spot, which is in the Western Ghats. So with respect to frogs that we captured the first time and we recaptured again, we did the swabbing of their skin. We swabbed the skin to test for chytrid. And we find with the dotted line basically represents no change. That is, if they were caught the first time and then recaptured on the y-axis, what is the number of the load of BD? If there is no change, they would lie on this dotted line. But you tend to see more dots on the lower side of the line than on the upper side of the line, which essentially means that if they were captured with a certain load the first time, and if you did find the same frog after a few weeks or months, you would find them with lesser load. And this is consistent across different species here. So the skipper frog, the amboli, jumping frog, the Indosilvirana, and the jog night frog. So all of them have similar patterns. So what's happening is that their survival is not affected by the amount of BD that is found. It is not impacting them much in terms of their survival, but there might be other impacts. And we know that because when we do capture these frogs, we collect them from a certain site in the stream and we go back and release them in the same site in the same night. And then when we capture them again, we note the distance that they have moved from the previous time they were captured. So when we looked at that, we found that frogs that had higher loads of infection did not move much. So there is some physiological impact of subclinical infections. We are talking about subclinical infections. Clinical manifestation of a disease can be that the animal shows some lesions on the skin and is not able to perform its physiological activities, and then it dies. But here they don't show any of those symptoms. They seem to be okay, but there is some subclinical impact that we are seeing with these frogs. So when we reconstruct the possible trajectory of frogs in those streams and how BD plays out on these populations, there could be a situation where a frog over time remains uninfected, but sometimes it might get infected and then go from low to moderate to high. But it has not been the case that they become so heavily infected that they die, or even if they get high infection, they don't die. So this situation is not true for the place where we work. So what is probably true is this, that they seem to be constantly shifting gear from low to high and high to low, or probably even clear of the infections and come back again with some infection. So because the BD is around in the population, it's just hopping from frog to frog. So they cannot resist the pathogen from getting onto the skin, but they can surely contain it on their body. That seems to be the message. So this also prompted us to examine was BD there at all in India before the pandemic emerged. So to just put you into perspective, the global trade on frogs has been the major cause of the spread of the disease, and Asia is supposed to be the origin of the BD fungus. Until 1985, 3,000 tons of frog legs were exported from India. And so probably there was some local consumption as well. So it's roughly 9,000 tons of frogs that were extirpated from natural populations from India. And India's major frog exports was an important revenue generator or a foreign exchange earner till that time. And then in 1985, there was a ban on frog-leg trade from India. So the shift happened to other parts of Asia, Bangladesh, Myanmar, Indonesia. All those places became important places to source the requirement of frog legs for the Western countries where it's eaten. So there was still exports happening from other parts of the Asia. So this led to the spread of the fungus. So our question was, what do we find signatures of BD in frogs in the collections historically from 1900? So we looked at almost a century, a period spanning over a century. And this work is being done by Yashwant, who's a PhD student in my lab. And we visited all the museums and collected samples. And then we looked at the different time periods are given here. And you can see the number of samples that we have, which are positive and negative. And you do find that there were a large proportion of positive samples from early 1900s, which means that the fungus was there. And it's not just that these samples are positive, many were positive, but we did find them with very high loads as well. So this study also points out that BD is not, frogs and BD have been dueling with each other for more than a century in India. And this whole situation that we see today is an outcome of this long period of interaction that has shaped some sort of host defenses, which we don't understand very well. So across taxonomic groups and geographic locations, we now know in South Asia, in the Indo-Malaya region, through museum samples that we have chytridiomycosis in frogs. So the lessons that we have learned through all this work that we did, that Western Ghats and Andaman Nicobar, though they have are mountains in different ways, they have very different histories. The Western Ghats are a result of Deccan volcanism, which goes back to more than 60 million years and 100 million years. So you have a long history of these mountains, while the Andaman and Nicobar mountains have probably been separated by less than 10 million years from the mainland. So there were a lot of sea level changes and volcanic impacts, which might have caused the mountains to subduct in the Andaman and Nicobar Islands. And the species diversity and endemicity is probably determined by the history of these mountains rather than their location in the geography right now. So mountaintop amphibians are going to be the most impacted due to climate change in the Western Ghats. And the same applies to littoral amphibians in the Andamans, because as I told you, they are the entry point into the island. They're the testing ground for any species that lands into the island. So if they survive the littoral forest, only then they will go and occupy the interior. So, and those forests are being put to a lot of change in the recent years. And the same applies to amphibians in the Western Ghats at the low elevation, because there's a complete change of land use there, and there's continued pressure on acquiring land which are natural, and important habitats for amphibians are being threatened due to this. Why there is a high rate of turnover in Western Ghats, we still don't have any much answers, because we don't understand how the populations survive in such narrow geographic ranges. How do they even survive such low numbers? How do they mate? How do they find mates? All those things are not so clear to us, because we don't know much about the natural history of many of the frogs. And the same applies to widespread species. Now that we know that there are some barriers for dispersal, and there are climatic variables that influence their activity, how do the widespread species maintain their populations? Do they breed every year? Do they move large distances? We are not sure, but they certainly move, that much we know. And for the islands, we still don't know how they disperse between islands. We know that frogs cannot tolerate saline conditions, but maybe there are rafts that help them move between islands, and they occupy these islands. So we need to understand how they disperse between islands better. And with respect to the BD infections, we now know that frog populations have a high prevalence, but with relatively low loads, and the infection is subclinical. And we now know that populations in our important hotspots of amphibian diversity can show stability in spite of very strong climate-driven events like heavy rainfall, cyclonic depressions. So they continue to remain stable. They can recover very fast, and this is attributed to their movement between the sites. And frog populations reduce BD infections and probably even tolerate some level of infection. And how do they do that is not very clear. As I told you, subclinical infections cannot be ignored. If chytrid is there, it is there and causing some impact. Though frogs may not die, they might show some effects, like they cannot escape from a predator, or they cannot breed successfully, or they cannot occupy areas to come at the right time and then successfully breed in natural conditions. So we have to take that into account and think about the drivers that are causing a continued decline of amphibians in this region. And what has happened in the past, we need to know that better. There seems to be some signatures of epizootic expansion of BD in early 1900s. We need to unravel it and understand it better. And the mechanisms that frogs have to resist and tolerate BD is also not understood. We need to do more work on that. Yeah, that's all. Thank you.
Thank you, Karthik. It was a splendid talk, and it really actually shows the way how critically you think and how critically you have made us think all the time. And probably we are at the last minute of our meetup. So, Kerry, if someone has any question, should they shoot the question, or they can write to us and Karthik can answer the question, and we can actually upload it on the web.
Yeah.
Let's do this. We've got about 3 minutes here. Sarbani, I'd like to know does Save the Frogs India have a social media channel? Not Instagram, because that's not ready yet. Is there a Facebook page or anything?
Yes, we have a
What is
Facebook
that?
page.
What's it called?
That's Save the Frogs India.
Okay. All right. Maybe you can chat in the URL of that.
Okay.
A short question, if you permit, I can ask. Hello.
Sir, please, sir.
Make it fast because we've only got 2 minutes
Very
left.
short question. Very short question. Karthik, can you tell me those frogs which were infected with BD, was there any difference in the call pattern? Did they make a different call, or they could not make the call, or the intensity of call or call pattern was different during the breeding season?
Yeah, it's an excellent question. I don't have data on that. We were capturing the frogs to swab them. So we don't know if they were calling or they called just before we captured. We are not sure. And we didn't record the calls. So yeah, I think something like that would be very important to do, considering that subclinical impacts are there.
All right, everyone out there, if you're looking for a project on subclinical impacts of chytrid infections, how does it impact their call ability? Let's see, everyone, we have a Facebook Save the Frogs India link down here. I'm also going to put in, we have a Discord channel. What is Discord? It's free. And the link is right there. It's a forum. You can ask questions. If you go in there, we have a channel that I believe is called India. Maybe we have one South Asia Meetups, but there should definitely be a channel in there. Look at the left sidebar, India. That I would say is the best place to ask your questions, because then it all goes in a permanently archived and easily found area of questions and answers and ideas and statements and discussions all about Indian amphibian conservation. So go sign up for that. If you don't yet have a Discord account, you'll need a Discord account, and put your questions in there.
And so on the last minute, if I can reintroduce Karthik, because that was not recorded earlier.
I think it was, but go ahead. Yeah, do it again, just in case.
Yeah, so Dr. Karthik Vasudevan is the chief scientist of CSIR cell of CCMB Hyderabad. And he is one of the leading herpetologists in India. And he basically needs no introduction, because he is knowledge hub himself. And we have learned a lot from him. And today it was just what we have seen is like the top of the iceberg.
Tip of the iceberg.
And,
Yeah.
tip of the iceberg. Yeah. So we are really grateful to him to come over here and share his insightful experience and research findings. And it's over to you, Kerry.
All right. Yes. Thank you, Kartik. Thanks, Sarbani, for getting everybody to show up. Everyone, please put a recurring calendar event into your digital calendar to show up on the last Saturday of every month at 7 p.m. IST. And I look forward to seeing you all again. I have one question for Kartik. I have been writing, though I'm not actually often writing it, but I started a paper called The Final Death of BD. Wouldn't that be cool if we got to hear about the final death of BD? And it's because I dislike, and I have always disliked, the term BD. Why? You mentioned binomial nomenclature. I don't know what Linnaeus would think of scientists being so lazy to say BD. It's not you, it's pervasive in the amphibian conservation community. So my one question is, in all your years talking about species and species names and binomial nomenclature, can you think of any other species that we refer to by its first letter of its genus and the first letter of its species, or is this unique?
I think it's pretty unique, but
okay.
yeah, right, because I think people are just too lazy.
Yeah, because in this day and age, look, if I type B, D into my computer, it just a capital B and then a D, it just pops out. We've got text expansion software to write it and to say B dendrobatitis. It doesn't take that much longer The problem is, I know from experience, an actual conservation problem occurs because BD, there's no relationship to chytrid in our minds. And I have been involved in chytrid related conservation to stop the importation of American bullfrogs into California, millions of them each year that carry a whole lot of chytrid infection. And the main government official tasked with leading that whole, the stakeholder meetings and things, it came to light one day that he had not made a connection between the term BD and chytrid.
Oh.
So I was like, oh, this is actually like impacting actual people who don't realize it. So anyway, I had never gotten around to asking anyone, like, does this happen in any other species? And if it doesn't, then I'm like, this, either we need to rename it officially, which I know nobody's going to do, because what would be the justification? Scientists are too lazy to say the long name, or, so anyway, do you have any thoughts on that?
No, absolutely right. I think we are feeding to very lazy biologists, just like, making like a baby food and, giving them information.
Yeah. So, but for everyone out there, it's not just Kartik. I see it all over the place. So maybe I will continue with that paper, and we'll see if it changes anything, or if it's too late.
No, it should. I think it's very important to do it, because as you said, it has impacts on what you can do for conservation. So yeah, you've clearly highlighted that.
All right, everybody. Thank you so much for being here. Thanks again to Kartik and Sarbani, and we will see you in a month. So enjoy your frog saving, everybody. Bye.
Bye.
Bye. Bye.